Hello, I'd like to welcome you to this webinar entitled Development of a Regulatory Grade Tool to Optimize Clinical Trial Design in New-Onset Type 1 Diabetes. My name is Joseph Hedrick, and I'm the Executive Director of the Type 1 Diabetes Consortium. Joining me today will be my colleagues, Kimberly Collins, Zihan Sui, and Paul Belmonte. First, I'd like to say a word or two about the Critical Path Institute. We are celebrating our 20th anniversary this year, and we have been a trusted not-for-profit generating solutions to advance and accelerate development of therapies for people with unmet needs. We provide a unique neutral environment for all the relevant stakeholders in the drug development ecosystem, and we leverage our core competencies to address unmet regulatory need across a wide range of indications. Today, we'll be talking about a tool developed by the Type 1 Diabetes Consortium. The Type 1 Diabetes Consortium was founded in 2017 and is working to accelerate the development of therapies for Type 1 diabetes. The interface that will be presented today is a pilot version. Ideally, it will enable clinical trial simulation using one of three univariate models: C-peptide, HbA1c, and insulin. As of today, only the C-peptide model has been submitted to the FDA's fit-for-purpose pathway. Importantly, the C-peptide model, while under review, has not yet been approved by the regulators and is subject to change based on FDA feedback. I want to say, finally, thank you to the members and the funders of the Type 1 Diabetes Consortium, and most importantly, to all of the Type 1 Diabetes trial participants who have made this work possible. Thank you.
Over 20 data sets with individual patient-level data were obtained from various sources. The raw data obtained were mapped into a relational data model and RDM using SDTM standards. Specific domains needed can be easily combined to create analysis subsets for modeling. This clinical trial simulation tool leverages three individual disease progression models developed to describe type 1 diabetes progression in individuals within the first 100 days of diagnosis. Now I will briefly describe each univariate model. C-peptide decline was best described using a sigmoidal Emax equation after data were log transformed. Significant predictors of C-peptide decline included baseline BMI, baseline age, and baseline C-peptide. Shown below are the visual predictive checks for the training and validation data sets based on an 80-20 split, indicating good predictive performance. The HbA1c model was best described using a combination of the sigmoidal Emax and exponential equation to account for natural disease progression and the initial dip and rebound in HbA1c observed within the first few months of diagnosis. HbA1c values were also log transformed, and only baseline age was observed to be a significant predictor of progression. Lastly, insulin use was best described using a quadratic model after data were harmonized into 3 to 7 day averages and square root transformed. Significant predictors for insulin use included baseline HbA1c, T1D duration at baseline, and age.
I'm glad to give you a brief introduction on the T1D clinical trial simulator we've developed. Currently, the tool includes three endpoints: C-peptide, HbA1c, and insulin. For each endpoint, users are required to provide information about the clinical trial design, patient characteristics, and assumed drug effect as input, which describes their target population. Then, users will get a disease trajectory plot and either a statistical power table or the number of patients, depending on user selection, as the output. Now, I'll guide you through the pilot GUI using C-peptide as an example. In the clinical trial design section, users can select study duration in months and endpoint assessment interval in months, which represents the duration between C-peptide assessments by adjusting the slider. Moving on to the patient characteristics section, you can select the baseline age interval, baseline 2-hour C-peptide AUC range, and baseline BMI score, Z-score range describing the population. This tool is based on the disease progression model mentioned by Kim, using a placebo model only dataset. And these three covariates are all significant, detected by that disease progression model. In the assumed drug effect section, there are two different calculation types, given sample size and given power. We'll talk about the given power type later, and now let's focus on given sample size type. Under this type, users need to provide total number of subjects in their trial, and these subjects will be randomly divided into two groups, treatment arm and placebo arm, according to the following user-provided randomization ratio. We have 7 selections here from treatment 1, placebo 5. In this case, the proportion of subjects in treatment arm is about 17% to treatment 5, placebo 1. And in this case, the proportion of subjects in treatment arm is about 83%. You also need to provide an estimated drug effect in percentage, which describes the increase of C-peptide from baseline. Finally, don't forget to set the number of simulations before you start your simulation. Now, I'm going to introduce the given power type. As you've seen here, all settings in clinical trial design and patient characteristics section are same as before. And in assumed drug effect section, the only difference compared to given sample size type is, in this case, you need to provide a desired power ranged from 0.1 to 0.99 instead of the number of subjects. And the Power GUI will calculate a recommended sample size in order to reach that power you provided for you. That's it, and thank you for listening.
Hello, my name is Pablo Monti. I'm the Type 1 Diabetes Consortium Scientific Director here at the Critical Path Institute. Today, first, I'm going to be talking about our data curation and repository that we do here at C-Path. In our work at the T1DC, we curate early onset T1D data to support clinical trial simulations and public access through our interactive GUI that we'll be describing here. Raw study data is mapped to a standardized structure based on a study data tabulation model with custom T1D specific domains to capture key biomarkers. Subject matter experts determine the most relevant variables, such as demographics, lab results, adverse events, to ensure consistency across studies. And then using our programming, we transform and store these data sets into a relational data model, or RDM, which aggregates multiple studies into a common format. The curation and RDM mapping code is maintained in the CPath GitLab repository with the final dataset existing as a structured .rmd file. And this curated dataset informs a clinical trial simulation tool. Again, I'll be describing that and walking you through how to use that today, enabling researchers like you to model disease progression and treatment responses. So in sum, this public-facing GUI makes the data more palatable, accessible to the user for exploration, and enabling our goal of transparency and engagement in T1D research and accelerating drug development. So now I'm going to be overviewing the interface with you, highlighting key GUI features like the input panel, simulation settings, and ultimately the results dashboard where you can view the output. So we've developed our clinical trial simulation tool to model disease progression and treatment effects for early onset T1D. Now, our initial univariate model regarding C-peptide has been submitted to the FDA. What you'll be able to use on this GUI is covering C-peptide, hemoglobin A1C, and insulin. These are individually univariate models. We are making progress toward a more multivariate advanced model that we will be submitting to the FDA later this year or early next year. So the tool features an intuitive GUI with two main sections. We have an input panel where your users can define the trial design, patient characteristics, and drug effects. And then the output panel, which provides real-time visualizations of the simulation results. So here you can see the input panel on our CTS tool regarding C-peptide first. I'll review. You can edit things like study duration in your clinical trial design and your endpoint assessment intervals in months. You can also modify your age. So let's do from 6 to 50. Your baseline C-peptide, we'll do 0.2 nanomoles per liter to 2.8. And then your BMI Z-score range. So you can have an assumed drug effect based on two options, either a given power or a given sample size. So we'll do a given power of 0.8 and have a treatment to placebo ratio of 2 to 1. And let's say we have an estimated drug effect of 75. So we'll go ahead and simulate. And as you can see here, the simulation outputs a graph with drug in pink and placebo in blue. Time in months is on the x-axis, and C-peptide AUC is on the y-axis with a 95% confidence interval. And you can see the drug does much better and compared to placebo. What is also outputted is the total number of subjects needed, so 165. Reverse, you could do a given sample size. So let's say, you do a little bit better with recruiting subjects. You still want to do a 2 to 1. Let's say 75% drug effect. Everything else remains the same. And so you have a similar chart here. And this time you have a statistical power output of 0.9%. So now we'll review hemoglobin A1C tab. Again, you can adjust your study duration. Let's go out to 2 years this time. Your patient age. We'll use a given power of 0.8 and a 2 to 1. And we'll use an estimated drug effect of 25%. So here you can see over time, the drug does better than placebo. Initially, you see this drop off here, indicating the time that HbA1c drops when patients start insulin when they're newly diagnosed. And the total subjects needed for this study is 147. Again, you can do the reverse and, let's do 300 again, 2 to 1 ratio. But this time, let's say the drug only has a 10% efficacy. And we'll simulate. Again, here you do really good in terms of statistical power. Total number of subjects, 300 as we inputted. And you have drug in pink and placebo in blue. Again, that initial drop off where patients start insulin. You can see over time, the drug does tend to go up towards 7. So you can play around with any of these parameters to fit your needs in designing your clinical trial or what you're interested in. So let's do insulin last. We'll do a 1.5 years for fun, a 2-month interval, adjust our age. And with insulin use, you can do a baseline hemoglobin A1C range. So here we'll do maybe 6.5 to 13. And we'll bring T1D duration up to 3 months. 15 days to 3 months. We want to give a power of 0.9 this time. 2-to-1 placebo. And let's have a 40% estimated drug effect and see what we get. So here you can see over time, you have a slightly less insulin use. This is in units per kg per day with a 95% confidence interval compared to placebo. And your total number of subjects needed is 837. Now let's say I don't have 837 patients. I only can do 300. Still want to do a 2-to-1. Let's keep it at 40 again. And let's see what happens. So again, here you can see a slight decrease in the amount of insulin needed with 300 subjects, but your power is very low. And so, you can play around with, well, maybe my drug is going to be have better efficacy, let's say 80%. And if you do that, the statistical power remains the same. And so, these are all parameters that you can play with. Again, this is based on existing data, and according to the data, it will provide projections of the total subjects you need, or statistical powers based on your input. So I hope you find this useful. It's a lot of fun to play around with. And I can't wait to share it with all of you. In summary, the T1D Consortium has developed an innovative clinical trial simulation tool designed to support drug development for a new onset T1D. The tool helps researchers, sponsors like you, design and analyze clinical trials using advanced disease progression models, ensuring confidence in regulatory submissions. At its core, the tool models key biomarkers: C-peptide, insulin use, and hemoglobin A1C to predict treatment effects, disease trajectories. The user-friendly GUI allows researchers to input clinical trial parameters, define patient characteristics, and simulate drug effects in real time. Outputs include our disease trajectory visualizations and statistical insights, aiding in trial design and decision-making. Again, our initial univariate model has been submitted to the FDA regarding C-peptide, and we are advancing a multivariate model for even greater predictive power. The tool integrates data from patient-level data sets curated through our programming into a standardized relational data model, or RDM. The structured data set ensures consistency across the studies, and while the tool does not provide direct access to raw data, it leverages aggregated insights to enhance research transparency. So providing this regulatory grade simulation platform, it's our hope that this tool will empower you, researchers, sponsors, and regulators to explore potential therapies with greater precision in T1D, and with the goal to accelerate the development of new treatments for type 1 diabetes. Thank you very much for joining us today. We hope this webinar has provided an understanding of how the T1DC clinical trial simulation tool can support your research. For further questions or a one-on-one demonstration, please reach out to our team or visit the T1D Consortium website.